I think hopefully you're great. So today I want to talk about a few different algorithms that we're going to use to study protein structure. See if I can get this. There we go. And I want to continue from where we left off last day. And so we were talking about some of the early algorithms to predict protein structure and do manipulations of protein structure. And those were mostly relied on energy functions. And that was kind of all we had before the advent of deep learning. So these physics-based methods using energy functions could do a lot. So we had some structure prediction. We could do docking of different proteins together. We could take small molecules, for example, drugs, dock them into the active site of a protein and try to figure out how could we modify that drug to make it fit better? How could we make it a better drug? Molecular dynamics is another function where we can take a protein, and if we understand the forces that drive that protein, then we can simulate what it does over time and get into the dynamics and look at how these proteins change and interact with other proteins. We can predict the effect of different mutations. Those might be mutations that we see in genetic data. And one of the most exciting applications, and one that we'll kind of briefly go over today, is protein design. So if we really understand protein structure, can we hypothesize protein structure and then figure out how to use this programming language of DNA and biology to make that structure, right? And if these proteins sort of carry out all the functions in the cell, can we rationally design new proteins for new functions, maybe functions that don't exist in nature? Okay, so these energy functions generally have a few different terms. There's a number of different energy functions that are widely in use. The main ones are going to be electrostatics. So just looking at how different charges interact with each other via Coulomb's law. We'll look at van der Waals forces, hydrogen bonds, and then proteins really do what they do, and they have the shape that they have because they're in water. They really only work in water. And so those interactions of proteins with water accounts for a lot of the energy and the stability of proteins. Okay, so electrostatics, at this point, probably you guys have all seen Coulomb's law. So Coulomb's law just says that the energy of two charged particles interacting is related to some constant times the amount of charge on each of those particles. In this case, when we're looking at proteins, we're usually looking at individual atoms, and so often we're just looking at a unit charge, right? So we've got a constant, and then it's divided by the distance between those particles, right? So the force is divided by the distance squared, but the energy is divided by the distance. And then if we have two charges in a vacuum, that's where you can just plug in numbers from some table that you look up. That's generally not the case in protein structure. We either have two charges that are separated by some amino acids in the protein, or we have two charges that are separated from each other in solution. And so there might be water in between them. And so the dielectric constant is important there. So those water molecules will rearrange themselves, and they'll tend to dampen out the effect of two charges on each other, whether they're kind of attractive or repulsive, right? So if we have two positive charges near each other, then maybe that oxygen of the water is going to get between them, and it's going to screen that charge a little bit, and it's going to make it a lot lower in energy to bring those positive charges together. So in water, the dielectric constant reduces the energy by a factor of 80, which is huge. And in a protein, you can expect that those side chains and things like that are going to reduce the interaction by a factor of anywhere between 2 and 10. Or if the charges are very close to each other, then it might not be reduced at all. Okay. So if you're looking at structure, let's say in PyMol, what I like to do, because a lot of what we're going to be talking about is using deep learning to calculate these structures and dynamics and things like that. So we're not going to be directly using these energy functions, but I think it's helpful when you're looking at structures to have a sense of, how strong is this interaction. So the simple method that I like to use for doing that is comparing it with something called the Bjerrum length. I think that's how that's pronounced. I've never actually heard anyone pronounce it. I've only seen it in textbooks. But the Bjerrum length is basically at what distance between two charges is the interaction between them equal to about 2.5 kilojoules per mole. And 2.5 kilojoules per mole is a really special number in biology because it's equal to KT or RT, where R is the universal gas constant. And it tells you how much thermal noise there is. And so at roughly 298 Kelvin, where all the biology in our body is taking place, you have water molecules that are constantly bumping into you and things like that. So if you have an interaction that's about 2.5 kilojoules per mole, attractive or repulsive, then you can expect to see fluctuations just due to thermal noise, just due to those water molecules kind of bumping into it, right? So KT is a special number. It's equal to 2.5 kilojoules per mole, which is really convenient. And in water, the Bjerrum length is 7 angstroms. So a carbon-carbon bond is about 1.5 angstroms. Okay, so if two charges are separated by 7 angstroms in water, then those interactions are just barely above the level that you would see from random thermal fluctuations. Okay, so that's not a very tightly bound interaction. Okay. So if for example we had two charges at 14 angstroms apart, then we'd know, well, the energetic attraction between them is just going to be half of what it is at 7, right? So it would be 1.25. And so that's a simple way to just kind of look at these structures and have a sense for how strong these interactions are. Okay. The next force is kind of the van der Waals attraction force, or together with the repulsive force is often called the Lennard-Jones potential. And if we look at it, what it's saying is that there's a distance. So at a large distance, like we were talking about for electrostatics, let's say 7 angstroms is where the electrostatic force is. It's just barely above the thermal fluctuations that you'd normally see. The Lennard-Jones potential, the amount of energy that we get from these van der Waals forces, it's almost 0. Okay, so it's not very strong at all. And then what you see is that when two atoms get really close to each other, then there's an attractive force between them. And this is part of what's really driving protein folding, is the protein folds into this 3-dimensional shape where those side chains fit together like a jigsaw puzzle, right? And when the surface of one atom is really close to the surface of another atom, there's this dip in energy. And so that's why they have to be like really well packed. And that's why you don't see proteins that have enormous holes in the center. Okay, unless those holes are filled with water or something else, because water can get in and also make those van der Waals forces. There's so the van der Waals forces are attractive, and it comes from the fact that we do have these thermal fluctuations just from water. And what'll happen is the nucleus of an atom will move with respect to its electron cloud, or probably the other way around, and that creates a small dipole. And that dipole is going to put a force on the atom next to it. And that atom is going to respond by moving its electron cloud in the opposite direction. And so then you'll have this dipole-dipole interaction where one is caused by thermal fluctuation and it induces an attractive dipole in the other atom. And those two atoms have to be really close for this to happen, and it creates this net force. But when two atoms get very close together, then those electron clouds are going to overlap, and that's excluded by the Pauli exclusion principle, and so the energy of that is going to be extremely high, and we really don't ever see that. So we can think of that as almost like a hard shell interaction. Okay, so that's the Lennard-Jones potential, and it reaches a minimum when two atoms are just barely separated. One of the most important forces in protein folding, and certainly in protein secondary structure, so how the amino acids are going to interact with the amino acids that they're directly bonded to, is hydrogen bonding. And so hydrogen bonds, we sometimes approximate them by thinking there's a partial electrostatic charge on this positive on one atom and a partial negative on the other. But they're actually quite strong, and it's really a quantum mechanical effect. And so for hydrogen bonds, we tend to evaluate those really empirically based on the statistics of how often we see those types of bonds in different protein structures. And so they're similar because they're like a quantum mechanical entity. The electrons are kind of shared between the donor and acceptor. They're a lot stronger than just a standard electrostatic interaction. They're more like a covalent bond, though they're maybe an order of magnitude weaker, so about 5 to 10 kilojoules per mole. We can also approximate the fact that some of the atoms that are bonded together, they tend to be bonded at a certain distance from each other. We know what are the preferred angles for different molecules, and all of those can be stretched and bent a little bit. And then there's those rotatable bonds that we talked about the other day, where I said we can approximate them as having 3 different states. Well, we can move them into a less favorable state. We just have to pay some energy to do that. Okay, and so a lot of energy functions will have potentials that help us get the bonded atom configurations correct. Okay, so some popular energy functions that are used are amber, charm, gromos, OPLS. These are collections of different terms in the energy function that cover everything we just talked about, kind of in one package. And so each of these has been parameterized to give optimal performance in different situations. And so a lot of these are used for molecular dynamics or protein structure prediction or refinement. These can be used with explicit waters, where, for example, in molecular dynamics, we'll just put a protein in a giant box of, thousands of water molecules, and we'll let the water molecules move around and interact with the protein according to the energy function. Or we can use implicit water models. And those implicit water models really get at the hydrophobic effect. And so for the hydrophobic effect, What we know is it can be approximated by looking at how much of the protein, especially the hydrophobic areas of the protein, are exposed to the solvent. And so for that, we can imagine, here I'm showing a cross section, but you could imagine taking this cross section through the Z plane and calculating a surface this way, where we take a water molecule and we sort of roll it around our structure, and we get we get that contour of, like what is the area of the surface. And so the hydrophobic effect, empirically, we know it's proportional to the amount of surface that is exposed to water. And so that's mostly an entropic effect, but we can treat it as in the same units as energies. Okay. So what do I mean by an entropic effect? Well, an entropic effect is really comparing two different states of a system, where in one state there might be multiple configurations, and in another state, let's say there's only one configuration. And so we could imagine that, let's say I had a really large box, and I put a particle in it, or I put a few particles in it, and I say, how many places are there to go in this really large box? Well, there's a lot, okay? And then you could imagine I take a really small box and I say, well, how many places are there to fit in this one? And let's say the first box is 100 times as big as the second box. And I were to put a little hose, connecting the first box to the second box. And I said, look, this box is roughly 100 times bigger than this one. How much time do you think any given particle is going to spend in the big box compared to the other one if it's just moving around randomly? Well, it's going to spend probably like 99% of its time in the big box, right? So we have a sense that the big box has 100 times, or let's say 99 times as much space as the small one. And so there's 99 times more configurations for the molecule in the big box compared to the small box. But now I could ask you the same thing, and I could say, okay, what if I took two boxes and they're equal size, okay? And I take one box and I bring it up to the top of Mount Everest, and then I take the other box and I bring it all the way down to sea level. And now my particle, well, I know it's going to spend more time at sea level, right? Because there's this like gravitational potential that's like pulling all the particles down, right? We know the atmosphere is a lot denser at sea level than it is at the top of Mount Everest. So I have this sense that like the bigger the box is, the more likely the particle is to go there. And the lower in potential energy, the more likely the particle is to go there. And what I'm missing is how do I combine those two things together? How do I convert from basically from one to the other? Okay, so that's kind of the central question of thermodynamics and statistical mechanics. And I'm gonna show you the formula. And so some of you may have seen this before, but this is how we do it. And this is the connection between energies and numbers of configurations. And at a deeper level, it's a connection between probabilities and a different kind of unit, right? Something that that's more like an energy. Okay, so when we think of the terms in the energy functions that I talked about, we think about the van der Waals, Leonard-Jones potential. We think about the electrostatic potential. We think about the solvent effects. Those are all in units of energy, in units of kilojoules per mole that come out of those functions. And what we do for each individual term to get the total energy is we just add it, right? And when we think about probabilities, if I say, you know, what's the probability of the particle being over here relative to over here? Let's say it's a half. And now I say, what's the probability that the molecule is oriented up versus oriented down? It's a half. Okay, so then what's the probability that the molecule is over here on this side and oriented up? It's a quarter, right? So we multiplied the probabilities. So the energies we add, the probabilities we multiply. So the natural relationship between the two is that one is a natural log of another, or to say it a different way, one is an exponent of the other, right? So the Boltzmann distribution tells us that if we have two different states a particle can be in, and the difference between those states, we can quantify in terms of energy, right? In terms of the number of kilojoules per mole. Then we can calculate how, you know, what's the relative log likelihood of observing the particle in one state versus another. Okay, so that's captured in this formula, and that energy it comes in some units, but we want it in really natural units. And so it turns out that the result of, you know, statistical mechanics is that we can compute, we know how to do that conversion. We know how to put it in natural units. So we can just exponentiate the energy. And the way we do that is we take the energy and we divide it by KT. And I already told you that for all the purposes we're going to look at in this class, KT is 2.5 kilojoules per mole. Okay, so if I have a particle that can be at energy, let's say 0, or a particle that can be at energy 10, well, then the difference between those two is going to be e to the fourth, right? I'll have e to the fourth times as many observations of the particle in energy level 0 as energy level 10. Okay, and importantly, there's a negative in the exponent, right? Because things are going to lower energy. Okay, so I want you to think about this for just a minute. Let's say a protein now has two different states, and this might be related to its function. So the protein can be open or the protein can be closed. And let's say the protein generally prefers to be closed. So the open configuration is 5 kilojoules per mole higher than the closed configuration. What fraction of protein molecules are going to be found in the open state? I'll just take a minute and just like do the calculation. Are you guys close to an answer? 11.9. Let's see. That sounds about right. Let's go through the logic. So it's 5 kilojoules per mole. So that's 2 KT, right? So it's going to be e to the minus 2. That's going to be the you know, some relative amount that's in the open configuration. So it's relative to what? The closed configuration. So that's just going to be 1, right? We'll call that 1. So then my ratio, my ratio is going to be e to the minus 2 over, well, what's the total number of configurations? It's going to be 1 plus e to the minus 2, right? Yeah. So e to the minus 2, I don't know. It's like maybe about like a tenth, 10%, right? So that looks like about 90%. Yeah. Okay. And so if we look at that formula, that's exactly what we put here for the Boltzmann distribution, right? We have the that relative energy for that particular state on top, and then we have to normalize it by looking at all the different states and dividing by those. Okay. All right, so now I want to go back to the protein structure that we looked at last day. And so that was the lac repressor, right? And what I didn't tell you is that, well, the lac repressor is also known to form higher order structures. So we know it forms dimers, but it'll actually form tetramers. And in the lac operon, we can see it forming tetramers where it'll bind one operator site called O1, and then it'll bind a different operator site. And those two versions of the protein will be bound together. And so we have these two palindromic sites that are near each other. And the 4 copies of the individual protein chain are binding these 2 copies of the DNA. And there's a region of the protein that we can see here that's important for the tetramerization. Okay. All right. So now I want to give you a slightly harder question. This one doesn't require any computation. It just requires logic based on what we've just talked about. And so our lac-I protein has, in fact, not just one additional binding site, O2. It also has another binding site, O3. And so, in that lac operon, we've got the O1 site, the O2 site, and the O3 site. And what we usually see is the lac repressor is going to bind as a tetramer to O1 and O2, or it's going to bind as a tetramer to O1 and O3. So there's 2 ways to do it, okay? And because there's 2 ways to do it, that means that, and we're going to assume that like binding to O2 and binding to O3 is about equal in energy, okay? But because there's 2 ways to do it, now that repressor is going to be bound twice as often, because there's a couple of different ways to for that to happen. Okay, so if we were to mutate one of those sites, let's say O3, we're going to mutate all the base pairs so that it no longer binds, and there's only one site. So there's O1 and then there's just O2 where it can bind. And what I want to do is I want to make sure that when I get rid of this other site, O3, that the lac protein is still bound just as often to the promoter and to the lac operon. So I'm going to want to do something to the O2 site to make it a better binder, right? I want the energy to go up or down. Down. I want the energy to go down, right? By how much do I need the energy to go down to compensate for the fact that previously the protein had 2 ways to bind? So this is the conversion. This is the conversion that I want you guys to just have be part of your intuition about thinking about molecules. So a number of configurations or a probability is related to an energy by a natural log. So what we need to do is we need to lower the binding energy to the O2 site by the natural log of 2 times some constant, which is KT, which is 2.5 kilojoules per mole. That's all we ever have to worry about. Does that make sense? Okay, so how could I go about strengthening that interaction between LAC-I and the O2 site? Yeah. Yeah. So I like that idea. I could go to the those residues and we look at them. There were like 3 that were in the major groove and they were touching the DNA, and we could say, is there a better amino acid that I could put here that would bind that site more strongly? The only problem with that is, well, if I change those amino acids on the lac repressor, then I'm gonna change how it binds to the O1 site. So instead, I would probably look at the base pairs in the O2 site and make sure that and see if there was a change I could make there that would change how they interacted with those residues from the lac repressor. Okay. All right. So now we've got a background on kind of the physics of protein structure. And I want to switch gears and get into the algorithms of protein structure. And I want to spend a lot of time today talking about AlphaFold, just because it's one of the most exciting achievements in the field. And AlphaFold is a deep neural network approach to predicting protein structure from sequence. And even though AlphaFold 1 used some of these energy functions, AlphaFold 2 doesn't use any of them. It really just relies on deep learning. But we're going to see that deep learning itself shares a lot of mathematics and shares a lot of intuition with statistical mechanics, which is what we're using to interpret how energies interact with number of configurations and things like that. So my goal for today is not to have you guys walk away with like a deep understanding of AlphaFold. AlphaFold is actually enormously complex, but what I want to you guys to walk away with is an appreciation for what's the information that goes into AlphaFold that makes it work? How is that information passed around the neural network? Okay. And how do we take something like a protein sequence that's the input to AlphaFold and encode it in a way that makes it a suitable substrate for a neural network? Okay, so that's kind of the goal. Next day, we're going to talk about language models, protein language models, and in particular, transformer-based architectures. And we're going to go really pretty far in detail, and I'm going to want you guys to walk away with a good understanding of how to build a language model, how to build a protein language model. Today we're just going for kind of an overview. Okay, so why are we talking about AlphaFold? Well, I mentioned earlier the protein folding problem has been around for decades and decades. And at the critical assessment of protein structure prediction, CASP 14, the organizers declared that AlphaFold 2 was a solution to the protein folding problem. So it routinely often gives structures that are comparable in accuracy to experimentally solved structures, and that was really the goal of the protein folding challenge. So this is a figure from the 2021 Nature paper when AlphaFold 2 came out. And so kind of the first panel shows the RMSD. Remember, that's the root mean square deviation of the C alpha atoms of the predicted structure versus the experimentally determined structure. For AlphaFold, it's shown in dark blue, and for all the other competing methods, it's shown in light blue. So just to show that, AlphaFold is blowing everybody else out of the water. But if we look at the structures, they're really, really impressive. So you can see the first structure, it's almost a perfect overlay. So AlphaFold is in blue and the experimental structure is in green, and they fit, almost identically on top of each other. And certainly like a protein at room temperature in solution probably has a lot of wiggle to it anyway. So, I don't even know how meaningful those differences are. But AlphaFold is kind of trained to predict the crystal structure of proteins, and so it does that quite well. There's a lot of I think weird things that happen in AlphaFold, and so that next panel C, shows an AlphaFold prediction for a protein compared to the experiment. And you can see they're pretty much on top of each other. But that black ball in the middle is a zinc ion. And so in the experimentally determined structure, you have these histidines that are binding that zinc ion from different angles. AlphaFold doesn't model the zinc ion, but it's really clear that based on the sequences that go in, AlphaFold knows the zinc ion is there, right? It has those histidines kind of cradling an ion of the of the right radius and things like that. So that's really impressive. And then when we look at AlphaFold and how it agrees with experiment on very large proteins, it also agrees quite well. So this is just really hugely exciting, and I think it opens the door to a lot of possibilities that we haven't even realized yet. So being able to have a structure for all the proteins encoded by genes in a particular genome, we have an intuitive sense that this is going to be really useful. Like there's a lot we could do with that. And the exciting part is kind of up to you guys, because we don't really know how to take advantage of that quite yet. And so, even if this is a solved problem, it's certainly not solved in all cases. But even if it were, the exciting part of what do we do with these models is totally open. Okay, and that's of course where it's going to have its biggest impact. Okay, so I said AlphaFold is a deep neural net. I know Professor Kellis talked about neural nets. I want to just give you guys a quick refresher. What is a neural net? Well, in a neural net, we're going to have some input in that that's generally a series of numbers, right? And then from that input, we're going to generate a series of activations at the next layer in the neural net. So what I'm showing here is a really simple feed-forward neural net where input enters at the left, and that's just going to be a series of numbers. So we can think of it as a vector. Those are going to get transformed to another series of numbers in that middle layer, the hidden layer. And then it's going to get converted to another series of numbers that is usually has a special form, and that's the output. Okay. So all of the circles on that diagram represent individual numbers. We're going to call those neurons. And those neurons are going to, in general, take some input and then produce some output. And the output we'll call an activation. Now, what's connecting one layer to the next? It's a series of weights. So the first, the top neuron in our hidden layer, that's kind of the, we'll think of the input as something separate. And so the first layer of our neural net is going to be that layer in the middle, the blue one that says hidden. And if we look at that first neuron, it has arrows going into it from each of the nodes in the previous layer, each of the nodes in the input. And each of those arrows is a weight, and it's a linear weight, okay? And so it's just a scalar that we're going to multiply the value in the previous layer by to and then add them together to get the activation in that hidden layer. So if we think about it, if we have in this case we have three input neurons, and we have 4 neurons in the next layer, and we're gonna generate those four values by multiplying each of the inputs by a weight and adding them together. That's just a matrix multiply, right? And so all of those weights that separate my the different layers in my neural net, those are just matrices, okay? And in addition, I can have a bias, and so I can take the weighted values from the previous layer, and then I might have a bias for each of those neurons. So I'll just have another number that I can add kind of optionally. Okay. So if I know my weights, which are a matrix, and my biases, which are a vector, then that really defines one layer of my neural network. Okay. So where do I get the weights and biases? Well, the weights and biases, they're just parameters in my model. And so what I'm going to do is I'm going to have a set of inputs and a set of outputs that I want to see on the other side. And I'm going to optimize those parameters until, when I put these inputs in, I get the outputs that I expect to see. Okay. And later we'll talk about the different algorithms that we use to do that optimization. Okay, so there's one more component to each of these layers that we haven't talked about, and that's the nonlinear activation. So if we think about the weights as being a matrix, and we can ignore the biases for a moment. If we think of the weights as a matrix, and I'm telling you that you can go from one layer of my neural net to the other by a matrix multiplication, well, then if I have 10 layers in my neural net, then I have 10 matrices. Why wouldn't I just multiply those matrices together in the beginning, a priori, and then take the resulting matrix that came from that computation and use that to represent my whole network, right? So in a sense, if all I have are these linear transformations, then I can only ever have really one layer, one matrix multiply in my neural net. Okay. So if I want to have more information stored in that network, and I want to be able to answer more complicated problems, then I need to do something else. I need a nonlinearity. And so what type of problems does that let me answer? Well, for example, if I wanted to encode a function like exclusive OR, right? So exclusive OR of x and y, if it's x is true and y is false, it's true, or vice versa. But if they're both true, then I want to map to false. There's no way I can do that with a linear model, right? However, if I have a model like the one shown, where that middle layer is nonlinear, so if the value coming in is below the threshold shown, then it's going to output 0. And if it's above the threshold shown, then it's going to output 1. Now I can actually create my exclusive OR gate, because if my input X and Y, if they're both 1, then it's going to trigger the first gate on the left. They're going to add together, and so they're going to trigger the gate in the middle. They're going to add to 2. They're going to trigger the gate on the right, and so all those gates, are basically going to fire, and then they're going to emit the number 1, and then you can see the weights coming out of that. The middle gate is going to get multiplied by -2. The gates on either side are multiplied by 1, and so they cancel each other out, and it's going to give me that 0, right? Okay, so having these nonlinearities allows me to answer more interesting problems, and it allows me to store more parameters in my model with every additional layer. And so this is a very simple nonlinearity. We had a threshold. If it's below the threshold, it's 0. If it's above, it's 1. But in practice, we're going to see different nonlinear activation functions. So ReLU is a common one. ReLU is basically just saying, gosh, if the number that ends up after adding all the weights and biases is less than 0, we're just going to call it 0. But if it's greater than 0, we're going to let the value pass through unchanged. People also use sigmoid function and tanh. But ReLU and different versions of that are kind of very popular because they're like extremely fast to compute and seem to perform as well as other things. Now, if we talk about the output of our neural net, the output of a neural net is usually thought of as a vector of logits. And we can think of logits as log likelihoods, right? And I think this actually has a beautiful connection to the physical problems that we were just talking about. And so what we generally do when we have an output of logits is we use the Softmax function, which converts logits, and kind of I like to think of everything in the intermediate layers as well, just everything in my neural network where I'm applying these linear transformations, I like to think of as logits, or having the units of log likelihood, and Softmax converts those to probabilities. How does Softmax do that? Well, this should look really familiar to you guys. It basically uses the Boltzmann distribution, okay? So it's saying, if those things coming out of the neural network we think of as energies, and here, instead of taking KT as 2.5 kilojoules per mole, we're just going to assume we've divided by that already, and KT is equal to 1, so we can just ignore it. Then what we should do is we should take the exponent of our logit and divide it in the denominator. So on the board here, I've got my 2 different states, right? But the output from my model, it might be 10 or 100 different states, or 1,000 different states. So I'm gonna exponentiate all of them, add them together, say, look, here's the total number of different states. What is the probability of each of these things, right? And I'm going to use the same mathematics that I would use if the numbers coming out of my neural net were energies, okay? All right, I think that's it's a beautiful connection, but I'll let you guys think about it. Okay, so now I want to talk about kind of the nuts and bolts of how AlphaFold 2 works. Let's talk about the input. There's multiple sequence alignment. There's something called a pair representation. They use series of computations called evoformers that are a little like transformers. Which we'll learn about in detail in next class. The output of that is a module that computes protein structures directly. And then AlphaFold 2 also outputs confidence measures. So we know on a residue by residue basis, which part of the protein structure should we actually trust, which is it most confident about. Okay, so here's the overall workflow from that 2021 paper. So we have an input sequence. The first thing we do with that sequence is we do a database search using a lot of the algorithms that Professor Kellis described to quickly identify other homologs of our protein. And then we build a multiple sequence alignment of those homologs. In practice, it's really just kind of a subset because those sequence alignments can be very, very large for some proteins. And then at the same time, we're going to build a matrix, which is the number of residues by the number of residues. And that matrix is going to represent the interaction between pairs of residues in the sequence. So for most proteins that we're predicting, that's the input. For some proteins, we're also going to, look in our database, not just a protein sequence, but we'll look in the database that we used the other day, RCSB, that has all the previously solved protein structures. And so if the sequence that we're trying to fold is homologous to a protein that has a known structure, we want to include that too, right? That makes sense. Interestingly, when they tested the model without putting in the additional templates from the protein database, the model performed almost equally as well. So at the end of the day, may have been useful in training and things like that. But you can also run it and expect good results even if you don't feed in the template, which I think is hugely impressive. Okay. So all of those data, they're separated in these two formats. So we have our multiple sequence alignment and a representation of that. We have our pairwise contact representation. Those get fed into this evoformer that iteratively passes information between those two representations. And then the output is passed to the structure module. And the structure module basically takes that information and then generates a 3D structure. And I think that structure is then fed back in, and the whole cycle is looped over 3 different times. Okay, so what's the input data? The input data is going to be a primary sequence, and then if it's available, a multiple sequence alignment of homologs, and that turns out to be fairly important, and then optionally a structural template. Okay, so what's the information that is coming out of the multiple sequence alignment? Well, here I'm giving you an example of a multiple sequence alignment in 5 different species. So let's say this is my protein sequence. There's a lot of different information here, right? So if we look at the the column of Ks that's completely conserved across all my sequences, and I've colored it red here, now I have a conserved position that I know is what's lysine, so it's positively charged. And one reasonable assumption is, if it's conserved and it's charged, then it's probably on the surface of the protein, or maybe it's in the active site, but that's also probably on the surface. Whereas if I have something like position 11 here in blue, tryptophan, and it's conserved across all my different species, well then maybe that is buried in the hydrophobic core, because I know those charged and polar residues want to be on the surface, and the hydrophobic residues want to be buried, because that minimizes the it minimizes their solvent accessible surface area, right? Okay, so that's good information. So I can look at the strength of conservation of that particular amino acid, and then use the tendency of that amino acid to be in different positions to inform my structure. The other really important piece of information comes from looking at the columns and looking at the interaction between the columns. So if we look at column 2 and column 15. Here we see positively and negatively charged residues that seem to be alternating, right? When position 2 is positively charged, position 15 is negatively charged. When position 2 is negatively charged, position 15 is positively charged. And so that's an example of a different kind of evolutionary information, co-evolutionary information, that suggests that, well, maybe residue 2 and residue 15 are actually near each other. They're interacting some way physically in the protein structure. Okay, so that's kind of information we're getting from the multiple sequence alignment. Then we have a pair representation, and so this we're going to generate by creating a matrix that is the length of our protein by the length of our protein. And I should say it's not just a matrix, it's a tensor. And so it has 3 dimensions, right? It has or 3 more than 3 dimensions. It has rows, columns, and then within each position in that matrix is a vector. And in this case, it's a vector of length 128. Why 128? Well, probably through empirical study, they determined that that was enough information to allow the model to learn what it needed to learn, right? Okay. So the interactions between any 2 residues then are encoded as a 128-dimensional vector. Okay. The MSA representation is similar. So one of the dimensions there is the length of the protein. The other is the number of species that are combined into the multiple sequence alignment. And then each position in that matrix is also not a scalar, it's a vector. And in this case, the vector is of length 32, because they probably empirically determined that 32 was sufficient to store the types of interactions that they needed to between different parts of that multiple sequence alignment. Okay, so I want to talk about this for a minute, because now we're kind of changing things from what we're used to. We're used to thinking about having these matrices with scalars and not tensors, and now we're putting these vectors in, and we're sort of hoping that these different vectors have all the information that we're going to need to predict structures at the end of the day. And so how is this done? Well, in general, we'll talk about embedding. And embedding is a way to take discrete data. So, for example, let's say an amino acid residue. And now we want to create a, so that's just, it's not even a scalar. It's really just like an integer, right? It's like a number from 1 to 20 or something. So now we want to take that and we want to expand it out to a vector of length 32 or 128 or something like that. and with continuously distributed real numbers. So this is something we're doing for both the MSA representation and the pair representation. And this is a technique that was certainly not first observed here. It's been used for quite a while, and especially in natural language processing. And so in natural language processing, one of the basic tools there is a program called Word2Vec. And in that program, it's basically not so much a program as a matrix. I can put in a word, and I could represent that word as a one-hot vector. And so if I have 50,000 words, and I want to represent the word cat, maybe cat is, word number 7. And so then I'd have a vector with 0, 0, 0, 0, 0, 0, 1, and then followed by, followed by all zeros down to 50,000, right? So that would be this this vector that represents the word cat, and it's not very useful. There's not much information in there, right? But I know that the word cat has a lot of meaning to it, and so what I'd rather have is a vector that's not dimension 50,000. But, maybe like 128 or 32 or something more reasonable. And I'd like that that new vector to not just be ones and zeros. I'll let it have, kind of any continuously distributed value. And I want vectors that that are similar to each other to be similar in their meaning, right? And so that's an embedding, and that's something we can learn, and we can represent an embedding as a matrix. In this case, if we wanted to convert all of our words into a 128 length vector, we'd have a matrix of size 50,000 by 128. And so how do we do that? Well, we learn what the different values in that matrix are by allowing them to be optimized as free parameters to solve some supervised learning problem. And if we're talking about natural language processing, the problem is usually, given the first half of a sentence, predict the next word. And in general, if we want meaningful embeddings, the key is to find the right supervised learning task that's going to generate embeddings that actually have some interesting structure. And so these embeddings often lead to surprising mathematical relationships between different concepts. And so I think this is, I just took this from the one of the Google pages, but I think it's probably Word2Vec. And so you can see that the relationship between man and woman versus king and queen is kind of the same. So if I were to take the vector for king, subtract the vector for man, and then add back in the vector for woman, the resulting vector would be awfully close to queen, right? And they have these interesting relationships. Or, I can look at the countries versus their capitals. And, there's this offset vector that lets you get from the country to the capital of that country. And so when I'm choosing the right task to optimize that matrix, I often get these sorts of relationships out by chance. And if I do the same thing, if I train a model, let's say a model to predict the next amino acid of a protein and make a similar map, I'm going to tend to get residues that have the same charge near each other, residues that are, about the same size and are hydrophobic are going to be all clustered together and things like that. Okay. Now we're getting into more of the guts of AlphaFold, and we'll post the slides so you guys can really dig in and try to understand what's going on here. It's actually not too complicated, although it it looks daunting at first. But what what I would like you guys to see is kind of how the features going in are turned into the input of the larger model. And we see that for a lot of these, we're just taking different features and maybe the embeddings of those features, and then we're either concatenating them together. So in the case of the MSA representation and the pair representation, they're just getting concatenated together. So they're different components of some much larger vector. Or if they tend to have the same meaning, we're adding them together. And so we're adding these vectors together, these vectors in a high dimensional space. And when I started learning this stuff, this was really confusing to me because I'm used to thinking about vectors in three-dimensional space. And if I'm standing here, and then from here, I go off in three-dimensional space in some direction, and I get to a new point. And then I choose another, I choose another direction. I get to a second point and a third point. After a very small number of steps, there's no way for me to backtrack like what steps I've taken, right? Like it doesn't make sense. Like once you take two vectors and you add them together, you have no idea, like you have no idea what vectors you added together to get to your final destination, right? In three dimensions, that's true. In these very high dimensional spaces, that's definitely not true. Okay, and so we can think about that, and let's imagine, and this will be relevant for our protein language models. The one we'll talk about, ESM2, is it's a 1280 dimensional space that we're taking each amino acid, single number, and converting it into this vector of length 1280. But let's imagine we have a vector of length 1024. Well, we could pick an arbitrary vector, okay, and let's say that's our signal, right? And we'll pick it to be 1 followed by 1023 zeros. Like that's our signal vector. And what I want to do is I want to take a bunch of different vectors in this space and add them together and see if I could tell that the place that I started was this vector, sort of pointing straight up, this 1 0 0 0 0 0. Okay, and so I'm going to say if I'm going to add some other vector in that I'm going to choose randomly, and if my cosine similarity of that vector is pointing in the direction of my signal vector, more than like 10% of the magnitude of the signal, then I'm going to say, hey, there's some interference, and now I can't really tell the signal anymore because it's being distorted, right? So what's the probability of that? Well, if I take a randomly chosen vector in a 1024 dimensional space, right? Then I can compute like what is how much of that vector is going to be pointing in that first dimension, and it turns out, well, it's going to be roughly the square root of 1024. It's going to be roughly 32. So on average, only a randomly chosen vector is only going to like interfere with that first one by about 3% of the length. And what's the probability if the standard dev is like 3% of the length? What's the probability that it crosses my threshold of being 10%? Well, it's like 3 standard deviations out. So it turns out that probability is very small. And so what this all means is I can take a lot of vectors and I can add them together, and then I can go back and just use just use a dot product or cosine similarity with the vectors I care about to figure out like what are all the things that that went into this, right? And so we'll see that in protein language models where we have these representations of 100 different amino acids in a protein chain. We can generate a single representation of the entire protein chain by like adding them all together, and then we can go back to that representation and say, oh, I wonder how many tryptophans are in here, and we can take a dot product with the with the embedding for tryptophan, and it'll approximately give us the right answer, and so it'll store a lot of information about kind of what's going on. Okay. So I think I covered that. The other thing that I want to talk about, so we have a sense that we can create these embeddings, and they're storing a lot of information about our protein in them. This becomes a lot more effective if we can pass information between different parts of the model. Okay, so we have a sequence of amino acids, and we want different amino acids to be able to share information with each other. And in particular, we want them to be able to share information in a way that helps us determine if those residues are interacting with each other. Okay, and so there's a number of ways that that's done in AlphaFold. And so they're using a mechanism that we're going to talk about next day called attention. But basically, what attention does is it allows us to take some data point, some vector in our sequence, and then compare it to like another data point in our sequence. So it allows us to take two different residues and decide, whether they have enough affinity for each other to share information. And the information is usually shared by adding them together. And then we can weight different parts of the vector differently in the addition, by means that we'll get into later. But the important thing that I want to point out here is it's done in kind of three different ways. And so in AlphaFold, the rows of our multiple sequence alignment are allowed to have attention with other rows, and that's influenced by the pair representation. So the pair representation, if we think about it as like the current three-dimensional geometry of the structure, it's influencing which rows of the MSA get to talk to each other. The columns of the multiple sequence alignment can also interact with each other. And then we can take those columns and compare them with each other. So we can identify these patterns where we had two different columns, and every time one is positively charged, the other is negatively charged, and vice versa. And we can look at those correlations, and then those interact with the pair representation matrix. Okay, so that was one of the big innovations of AlphaFold 2, is how these different representations get to share information in these evoformer blocks. And then how is the 3D structure stored in the pair representation? How do we even know that it's stored in there? Because that first slide I showed you about how we generate these in the first place was kind of gobbledygook, and there was we just sort of took all the data and kind of threw it in there, and we said, well, there's a lot of parameters in our neural net that get optimized to represent structure. How does that happen? Well, it happens in a few different ways, but mostly because one of the things that the model is being trained on is to predict the distances between those pairs of residues. So we have some supervised learning problem that's creating the weights in those matrices such that they have enough information that at the end of the day they can predict the distances between those different pairs. There's something else that's really unique to AlphaFold 2, as far as I know, that helps to enforce this. And that is that there's special passes where residue ij, that pair, and then residue jk, that pair, get to inform the pair interaction between I and k. And so they have these triplets. And by allowing those triplets to share information, they implicitly enforce the triangle equality, right? And the triangle equality says, the distance between I and j has to be less than or equal to the distance from I to k and then k to j, right? We know that geometrically that has to be true. And the way that they've structured how information is shared in the pair representation matrix helps to implicitly enforce that. Okay, so because we're training it to output the distances between residues, and it outputs actually a probability distribution of distances in a bunch of different bins. Because we're predicting that, we know that this matrix has enough information to store these distances. It's also storing the orientation, so the phi psi angles and also the orientation of the residues themselves and the direction that the backbone is moving in, and all these things are fit during the training process. At the end, after going through 48 different blocks of the transformations that we just saw, and each of those blocks has many parameters, and the parameters in each of those 48 blocks are different. So AlphaFold ends up having a huge number of parameters in the model. Then we get a final MSA representation and a final pair representation. And in AlphaFold 1, they would take this information, put it into an energy model, and then find a structure that kind of satisfied those constraints. In AlphaFold 2, they basically just put it into a neural net that directly spits out a protein structure. Okay, so that's kind of the walkthrough of AlphaFold 2. In addition to the structure, it's predicting the confidence of different positions. Yeah. Yeah. It is, but it doesn't. I mean, it doesn't super matter. Like I think it is because that's the most straightforward. But it doesn't really matter because one of the features that goes in is just its numerical position in the sequence. So that goes in, that number goes in, and it gets converted to like that embedding.
Innovations of the transformer architecture was the fact that you didn't have to put things serially. You could just throw in the position, and we're going to see that at the next lecture. So I think this is one of the advantages that as long as you know the matching between what position in the initial sequence on the left corresponds to what residue, you can just the algorithm can figure it out.
Yeah, and there's a number of places in the algorithm where they will explicitly put in the relative distance between the two pairs. So they actually compute that multiple times and put that information in. No, I mean the relative distance in the linear sequence. Yeah, yeah. Okay, so I'm going to stop there, and we'll continue with this, and then get into ESM and protein language models next day.
And recitation tomorrow. I know that PSET 1 is due today. Recitation tomorrow will walk you through PSET 2. If you guys are interested in having that tomorrow, raise your hands. Walk you through PSET 2. Awesome. So we'll have that tomorrow. Remember, submit your PSET 1 today. We're going to walk through PSET 2 tomorrow, and next Friday we're going to do another one of the metric sessions.
Good. Pause.
Stop.